Hi, this is Terry Kuzma at Penn State University. What we're doing today is taking a look at materials and nanotechnology. We're going to look at part number 4 of our presentation on nanoparticles for medical applications. So we're going to continue to take a look at liposomes, but not specifically for cancer, for other applications. Then a brief look at some metal nanoparticle applications. So what we can look at is that liposomes are not only approved for cancer applications, but other types of areas. And I'm not that familiar with these other types of applications, but what we reviewed is certainly this one down here for HIV-related Kaposi sarcoma. But they can also use it for influenza, so I don't know how that's done. I don't know how they'd use it for macular degeneration, et cetera, but you're more than welcome to take a look and do research on your own in these specific areas. So nanoparticles, they can also be used for tagging. So the nanoparticles themselves, because they could be either plasmons or quantum dots, we can adjust the size and get different colors. So we might want to track where a specific protein goes into the body, or multiple proteins. We can take specific proteins and tag them at different colors, and then at the same time watch these different colors associated with different proteins make their way around the cells. So quantum dots themselves are actually a very good tool for tagging in the body, because they're usually about 20 times brighter and a lot more stable than conventional phosphorescent dyes. Usually we would use nanoparticles or quantum dots on the order of 50 nanometers. We could potentially put things like that into a payload of a 300 nanometer liposome and be able to generate that within the body effectively. So we could also provide genes within the body. So we can provide therapeutic gene therapy with the conduit of the liposome being the material, the workhorse to deliver at the different areas. I don't know if this is actually approved in the United States. I think this still might be in the procedural stage and not open to general practice, but I'm not sure of that. So this is how genes would make their way through. Sometimes we can have endocytosis through the cell wall. Basically, the liposome is able to be engulfed by the wall and then deliver the payload directly into the cell matrix. So we can also look at, there's applications which are, I guess, common for nutritional supplements, such as encapsulating vitamin C in liposomes. So these tend to be sold, I think, over the counter. I don't know how these are sold as a food supplement, not necessarily as a therapeutic FDA-approved system. So I have my doubts on this, but it can be used in that way. I think some of my doubts are, the claims are, if you would take vitamin C in this particular mode, then you'd double your lifetime, etc. I think that's what I would term snake oil, a false sale. So liposomes themselves are biocompatible. They feature the same construction as cells. They're made out of a lipid bilayer. Liposomes can be delivered directly to the appropriate site by passive delivery, by ligands, or by local injection. And delivery methods conserve material and minimize side effects. And I think that's especially important. Liposomes can deliver both fat and water-soluble drugs, RNA and vitamins, and liposomes themselves are in widespread use. We can also look at another nanoparticle for medical applications, and this would be the metal nanoparticles. Since the size of nanoparticles are comparable to biomolecules, nanoparticles can overcome biological barriers. Nanoparticles themselves can be 100 to a 10,000th the size of a cell, and this will increase the uptake of this material within the cell, because the cell will think that the nanoparticle is a naturally occurring enzyme or an antibody, etc., or food. The shape of the nanoparticle also has impact, and generally, if we would look at cells, there's been quite a few studies that say that spherical objects are accepted a lot better than rods and triangles, etc., to the body. So gold is often used as the metal of choice because it's been shown that it's relatively inert to perform the work as a delivery system. Some people have issues with gold building up in the liver, for example, but a lot of times we have to look at drugs in it as far as there always will be side effects, and maybe the side effects from gold, if there are any, might be acceptable given that not using that particular treatment. So, but a problem, some people identify gold residing in the body for long periods of time and building up in the body. It could be bad, especially in the liver, but that hasn't been proved yet. Gold is expensive in the bulk naturally, but on a nanoscale, when we're using, nanometer size stuff, we really don't need that much. So the small volumes that are necessary for conventional treatments, really the bulk cost is really not important at all. Metal nanoparticles can be used to target and destroy disease cells. There are different functionalities of these particles to perform selected tasks. They can be used as tags to identify. They can be used as energy receptors to cook tumors, and they may be used as delivery platforms for drug or RNA delivery. Gold nanoparticles can be used as a marker to delineate between healthy tissue and tumors. In this role, gold nanoparticles are coated with a ligand that will preferentially adhere to tumor cells. These gold nanoparticles are grown to a specific size, so they'll exhibit plasmon resonance. When a light source is used to stimulate the plasmons, the surgeon can see where these gold nanoparticles are binding to a tumor, and then that way what he's going to do is minimize the trauma to the patient by cutting less of healthy tissue away and being able to resolve the cancer in more detail. So gold nanoparticles are considered, for the most part, relatively inert, so side effects are limited or non-existent. Gold nanoparticles are relatively non-reactive, so they can be coated with polyethyl glycol. So they're very good at moving through the body, and they appear to be stealthy, just as we discussed the liposome. Small nanoparticles are more apt to penetrate the tumor because they can passively slip through the blood vessels, as we recall as in the EPR effect. Gold nanoparticles are not good at keeping light in an excited state. So when illuminated, the light energy state can get off as heat. So basically what we can do is we can place gold within a tumor, apply some kind of light or an E&M wave that's transparent to the rest of the body, and what it's going to do is resonate on the gold particle, and if the gold particle is locally located in a tumor, what it's going to do is heat up that tumor and kill the tumor. We can also, with nanoparticles, apply antibodies, and these can be used to go to specific recognition domains within the body. So the selection of antibodies made so healthy cells have a low level of the receptor, then the gold nanoparticles will seek the cancer cells preferentially. By illuminating gold nanoparticles with light, they can give off heat to be estimated to be 70 to 80 degrees, and what this localized heating is going to do is kill those surrounding cells. So heating is based on the known optical plasmon resonance of gold in the near infrared region. This therapy is currently in phase 1 clinical trials for the treatment of refractory and/or recurrent head and neck cancer. Though highly effective for superficial tumors, near infrared is not really suited for tumors that are deeper in the body because the penetration depth of the near infrared is limited to about an inch or so. This severe limitation but is addressed with other nanoparticles that we'll take a look at later. So the answer to that is the nanoshell. So the nanoshells can actually use a different E&M segment of the spectrum, And what they're able to do is that E&M is transparent to the body, so therefore we can place the nanoshells deeper in the body and still be able to get this heat ablation. So blood and tissues will absorb most of the wavelengths of the material. However, wavelengths that are invisible to the eye, such certain ranges as the infrared, will penetrate through the human tissue with minimal absorption. The nanoshells are manufactured to capture the infrared heat and infrared waves and convert them to heat. The tuning itself is done in a nano shell as a silica core with a gold outer shell. So gold nanoshells, the key innovation with nanoshells is that we can tune their color from the visible using the dimensions of the core and the shell to differentiate regions of the spectrum. So the particles can be tuned to the near infrared region of the spectrum. Bioengineers call this the water window, because we're mostly made out of water, and the water is the most transparent in this region of light. So therefore we can have the light penetrate the tissue in as much as 10 centimeters, dependent on the tissue type. So that's good, because if we would have the tumor somewhere in the body, 10 centimeters is pretty far, that we could shine the light from the side, the back, the front of the body, and be able to get that energy to the point where it's needed to do the ablation. So this shows the tuning of the gold nanoshells, and basically what this researcher Naomi Hallis did down in Texas, she was able to change the relative size of the gold on the silica inner core, and she was able to change the resonance of that particular nanoshell. So this is what she was able to do, is by size and by shape, is able to shift the resonance of that material. So this professor of electrical and computer engineering at Rice University, Naomi Hallis, is the inventor of nanoparticles called the nanoshell. These are called oroshells, and they're being interrogated commercially, and they're in the FDA process, and they're doing research on that material as we speak. So quantum dots and plasmons have a wide variety of visual tagging for RNA and DNA, so we could also use these metal nanoparticles for that tagging area. We also use metal nanoparticles for tagging in sequencing of DNA, and we would use this in applications like the Affymetrix DNA lab on a chip, and this is the type of technology that they have marketed in the past. So nanoparticles can also use to prevent infection from bacteria, etc. So they're a biocide or an antibacterial. So what we can do, and this is a research, we'll take a look at a research paper here and some data. What people have done is they've coated parts like screws or prosthetic devices with a certain percentage of a nanoparticle. And the amount of silver content on the surface is shown here on the x-axis. So Titan is, I think, a titanium bare prosthetic material, such as a screw, a titanium screw, and it has 0 silver content on it. Then what we do is we apply 0.7% silver content on the surface, 3% or 4 or 9%, and then what they did is they studied that impact of that silver nanoparticle on the surface to this particular bacteria. And then what they did is they also took that silver at the same ratios, and they took a different type of bacterial infection agent, and they looked at its effectiveness there. So we can take a look at the amount of growth associated with, in proportion to the amount of silver on there. An issue with this, though, is what happens is the silver, as we place the silver on the prosthetic device, what it's going to do is it's going to inhibit cell adhesion. So while we place silver on there and it destroys the infection, what it's going to do is take longer for the patient to heal. So this gives us the relative magnitude of healing cell activity or protein activity on the material with the particular silver application. So what we would like to have is certainly a high cell activity, but this is shown at certain day periods. So basically what people do, they have to take a look at the ability to repress the infection and also the ability to repress cellular growth. And you have to find a sweet spot there to where you're repressing the infection and you're not repressing the growth that much. And so at that happy spot where those grafts intersect, that would be the dosage that you would use on your product. So I thought this was a good study because it shows both the positive and negative impacts of silver nanoparticle on prosthetic devices. And again, as a story, when you're trying to look for these cures or for the ability to prevent infection, a lot of times what you have to do is accept other negative attributes, like it's going to take longer to heal. But I think most people would say that the thing that you don't want to have at all is infection or failure of a prosthetic device, because often a second implant doesn't work at all. That's very difficult to get a second procedure to be done correctly. So you would probably rather be conservative and have the healing time be longer, but assured that we're not going to have catastrophic failure from an infection or some other non-ideal parameter. So wrap up in our conclusion here, biological processes themselves set the scale of material processing needs. So what we do to design our biomaterials or nanobio drugs, etc., or prosthetic devices, we actually have to look at how does the body react to these materials. And what we have to do is manufacture and make these biomaterials conducive to the body. So nanoparticles such as liposomes, gold nanoparticles, and nanoshells are really of the appropriate scale to interact on the cellular level. And certainly biotechnology is an exciting field and offers a lot of career paths and really good science in nanotechnology. So again, I hope you enjoyed this presentation, a very exciting field, and I look forward to talking to you more about other applications in nanomaterials in our next lecture. Take care.